Lathrop worked very hard to try and persuade me into taking a tour all the way to Mill Creek, and I will admit I felt like it was a little bit too far, but I bet you could contact him and he would be happy to arrange something for a group of folks who would like to see it. It's quite interesting to see the Mill Creek property. So for our concluding speaker for our silvicultural section, I'd like to bring up Dr. Lucy Kerhoulas. She's with Humboldt State University. She's a ecophysiologist and a new professor at the university, which is great. She got her career started at Humboldt State and then went away to northern Arizona and was able to return as a professor. So she's got a wide variety of interests around physiology, and I'm looking forward to her talk today. She's going to be talking about physiology and growth of redwood and Doug-fir planted after variable density retention. So, thank you, Lucy. Okay. Pointer, and then that's the forwarding back. Okay, great. Thanks. Yeah. Okay. Hi. Well, I feel like. Oh, yeah. Hi. Thank you for coming to hear my talk. I'm Lucy Kerhoulas, and today I'm going to be talking to you, as Jana said, about the physiology and growth of redwood and Douglas-fir seedlings following variable density retention treatments. So this talk, you might be wondering, okay, so why should we care about what's going on with the seedlings? And for two big reasons. In this, we're having a widespread forest mortality, largely drought-induced. And so on this map here is just a figure showing all of the forest mortality, particularly down in the southern Sierras. So as we have this widespread tree mortality happening, efforts for reforestation might be interested in what's going on with seedling establishment and the success of seedlings after planting. And then also just the timber industry here in Northern California, if you're going in and doing timber harvest and want to replant, especially in this warming and drying climate, it's an important question to consider how are our seedlings responding physiologically and also as far as their growth capacity. So those are sort of the big why questions about why you might be interested in these topics. This study that I'm going to tell you about today is just, it started out, it is a pilot study, so a 2-year pilot study, and it's been a real interesting experience working on it because it's been a big collaboration with the HSU Forestry Department. Almost all of the professors have been kind of involved in one way or another, as well as a lot of the students. So it took place at the LW Schatz Demonstration Tree Farm out in Maple Creek, which is about 25 miles east of here. Our study site is about 1,000 feet elevation, and it's a Doug-fir tan oak forest. And in 2014, in the fall of 2014, the silviculture class, the HSU silviculture class, went into this study site here at the Schatz Tree Farm and did a variable density retention treatments. So they had 5 different treatments done on 15 different 1/10 acre plots. So the whole study site, it was very small. This is just a little pilot study to see how this would work. And so the retention ranged from 0 to 100%, 0 being like a gap in the forest, and then 100% being a control where nothing was taken. And then in the spring of 2015, the forest restoration class went in and replanted this study site. And so they planted Douglas-fir seedlings and redwood seedlings from the John Steen Nursery up in McKinleyville. And then also that spring, shortly after planting, the HSU Silvics class, so a lot of undergraduate involvement, went and started doing some baseline growth and physiology measurements to these seedlings that were planted. And because the physiology measurements were going to be destructive samples we'd be taking from the seedlings, we ended up having to plant 3 what we dubbed sacrificial seedling patches that we could be harvesting from over the 2-year period for these measurements. So the 1, 2, 3 there are 3 sacrificial plots that were planted with seedlings from green diamond. And we planted again in these sacrificial plots, Doug-fir and redwood. And so in plot 1 there, that was a low retention site, so only 15% of the overstory retained. And then plot 2 was a control site, nothing was taken, so 100% retention, low light. And then plot number 3 there was our kind of middle of the road road, so a little less than half of the trees were retained with 45%. And then, yeah, so that was kind of the lay of the land as far as the study went. And so going into this, we started off with two main hypotheses. The first one having to do with the variable density retention treatments. We had hypothesized that the moderate retention treatment would sort of be the best as far as seedling success went, for a couple of different reasons. One being that you'd kind of be in that sweet spot of light and water. So you'd have enough light, the seedlings would be released from light competition, but there wouldn't be too much light that the seedlings sort of got fried in that first growing season of summer. And then also having to do with kind of water in this different treatments. In the really dense stands, so the high retention, there would be a lot of through-fall interception from the overstory, so maybe those plants might not get as much soil water recharge. And then in the really open, low retention treatments, it was hypothesized that there'd be a lot more direct solar radiation hitting the seedlings, increasing water stress, and then also a lot of solar radiation hitting the soil, so increasing evaporative loss of soil water. So hypothesized that the moderate treatment would be the best. And then the second main hypothesis was kind of the interspecific comparison, so looking at Redwood versus Douglas-fir and how these seedlings behaved. And we had hypothesized that redwood being off-site, it doesn't naturally occur here at this study site. We had hypothesized that it would not do as well as the Douglas-fir seedlings just because it's further inland. Maple Creek is further inland. Redwood doesn't occur here. It seemed that it would be too hot and too dry for the redwood seedlings to really do that well. So that's kind of going in. More specifically, we talked about kind of water stress, so hypothesizing that the redwoods would be more water stressed compared to Douglas-fir, suffer greater mortality compared to Douglas-fir, and as a result have reduced growth compared to Douglas-fir. So to test these hypotheses in the first growing season, the 2015 growing season, we took growth measurements, so just really simple measurements, basal diameter and height at the beginning of the growing season and then again at the end of the growing season. There's Wade Polda, one of the collaborators, he did his senior thesis for botany on helping with this project, so it was pretty cool to be a part of. And then to evaluate water stress, we did water potential measurements using a pressure chamber. And so those were kind of our methods for the first growing season. And what we found, just kind of one thing to note was throughout the 6-month growing season, if we think of the growing season May through October, August seemed to be, this is for both species pooled, August was the highest water stress. So in this figure here on the vertical axis is water potential, and the more negative the number, the more water stressed the plant was, the more tension was in the water column. And these are pre-dawn water potential measurements, so it's really fun to wake up early and go out and do field work with a headlamp. But you do get, this is considered sort of the most relaxed water status that the plant can have. So at night, presumably there's no stomatal conductance going on. You can have leaky stomatal conductance, but the plant can basically re-equilibrate with soil water. So in the pre-dawn, before the sun comes up and the plant sort of physiologically wakes up, that's the most relaxed that the plant will be all day. And the more relaxed the plant starts off the day, the more relaxed the plant will be at midday. So presumably having higher photosynthetic rates at midday when that there's make hay while the sun is shining kind of thing. So here we can see that for both species, this is both species pooled, August was the most stressful month, and then once September, October started to happen with shorter days, some more fog rolling in, water stress seemed to relax a little bit. The second thing I thought I'd point out today, just based on the 2015 findings, has to do with the treatment. So contrary to what we'd hypothesized, the low retention treatment across the entire growing season had significantly more relaxed water potential values at the pre-dawn compared to the other two treatments. And this was surprising. Again, I had hypothesized that the really intense thinning treatments that left very few trees there, that would just be really intense for the seedlings that were planted, but they actually seem to be faring the best across the entire growing season as far as their status went. So that was surprising to find. So the low retention being the white columns there. And again, pretty substantially reduced water potential values compared to the other two treatments. Regarding the interspecific comparison between Redwood and Douglas-fir, looking at water status, so again on the vertical axis having that pre-dawn water potential value, contrary to what I had hypothesized, the Redwood was actually significantly more relaxed as far as its water status went compared to the Douglas-fir. And this was really surprising to me. Again, Redwood not naturally occurring at this site, it being a little inland out of the fog belt. I thought that they would be more water stressed compared to the Douglas-fir. But across the entire first growing season, you can see these data show that the Redwood actually maintained a much more relaxed water potential value compared to the Douglas-fir. As far as growth went for that first growing season, so if we, as our growth metric, if we look at the change in basal diameter, here we have the 3 different variable density retention treatments on the x-axis, and then Redwood and Douglas-fir, so Redwood in white, Douglas-fir in gray. And you can see that Douglas-fir had greater growth in that first growing season than Redwood did. And this is, that was what we had hypothesized would happen, but it's kind of like head scratching, because given Redwood's water status, which was more relaxed, you would think that maybe Redwood would have grown more than Douglas-fir in that first growing season. But when we look at the growth data, Douglas-fir had more growth in that first growing season as seen as change in basal diameter compared to the Redwood. The other thing kind of interest here is the interspecific comparison, or the treatment comparison. So again, the low retention treatment had the most growth. The seedlings in that treatment experienced the greatest growth compared to the other 2 treatments. So again, kind of going with the previous talks in this session, that the more open the canopy after a thin, kind of the trees that are there, they seem to do better. And then the last piece of the story that I'll report on today is looking at mortality. So again, we see that in the low retention treatment, mortality was the lowest for both species. In the first year, none of the Douglas-fir seedlings died in the low retention treatment, but in the moderate and the high retention treatment, Douglas-fir had about 35% mortality, whereas Redwood didn't have as much mortality, maybe due to the fact that it had more relaxed water status, so it wasn't as water stressed as the Douglas-firs. So to kind of recap the picture here regarding the interspecific comparison, we have Redwood, which was was less water stressed than Douglas-fir for the first growing season. It had less mortality than Douglas-fir in 2 of the 3 plots for the first growing season, but yet it grew less than Douglas-fir, which is a little bit perplexing. So how do you make sense of that? Well, there are 2 kind of potential explanations that I can think of, and they're not mutually exclusive. So the first one is that Redwood is behaving more isohydrically than Douglas-fir. So isohydrically meaning that it likes to maintain a kind of even-keeled water status. So at early signs of water stress, the plant will close its stomata to conserve water and avoid potential cavitation and embolism in the vascular system. But the drawback to that is that there's no more CO2 coming into the leaf. So photosynthesis kind of is reduced or comes to a halt. And this, if this is indeed what's going on, would explain the relaxed water status of Redwood compared to Douglas-fir, because there's been stomatal closure to conserve water. And it would also explain the observed decrease in growth in Redwood compared to Douglas-fir, because there was reduced photosynthesis due to stomatal regulation. A second option to kind of make sense of the 2015 findings is that Redwood, and again not mutually exclusive, is that Redwood is investing more of its photoassimilates, so more of its sugars from photosynthesis, into below ground, so into its fine root production and or its mycorrhizal symbionts. And if this is in fact what's happening, this would explain the increased or a more relaxed water status of redwood compared to Douglas-fir. If there's more fine roots being produced and more mycorrhizal symbionts, that would allow for greater water uptake and more relaxed water status in redwood. It would also explain why above ground redwood showed less growth compared to the Douglas-fir, because more of the sugars are going below ground compared to in the stem for radial growth. So those, to get at kind of which of these two things is happening, or maybe potentially both of these things are happening, for the 2016 growing season, we added a couple more field measurements to our work. So in the 2016 growing season, which we're just about to wrap up at the end of October here, we continued with the growth measurement, so measured basal diameter and height of all of the seedlings at the beginning of the growing season, and then we'll do that again at the end of the growing season. We continued with our every other week water potential measurements out at the site for pre-dawn and midday. And then we added to that stomatal conductance measurements using a leaf pyrometer, a decagon leaf pyrometer. So this is kind of like the iPad of like plant phys toys. It's really great. It's really great for broadleaf species, but with enough finesse, with a certain amount of finesse and kind of attention to detail, you can do it on conifer leaves. So stomatal conductance measurements, and then also at the end of this growing season, I'll do a destructive harvest of all of the seedlings and evaluate biomass accumulation above and below ground on these seedlings to try to get at how carbon is being allocated, and maybe that can explain some of the differences in physiology and growth observed. So stay tuned for that. And I guess the big picture kind of take-home points, I know you guys have heard a lot of talks today and a lot of information, so if you can take anything away from this talk, what I'm seeing at this study site is that the low retention, so the heavy, thin silviculture treatment, seems to be the most promising for successful seedling establishment after planting. And then secondly, that redwood is doing really well at this inland site where it doesn't naturally occur. So this is sort of encouraging or boding well for redwood's ability to acclimate or adapt to the warming and drying climate, at least currently. I'd like to acknowledge my collaborators, Nick Kerhoulas, Wade Polda, Pascal Barrel, and the entire HSU Forestry Department, and then all of the HSU students who have helped on this study site. This research was funded by McIntyre Stennis money, and then also thank John Steen Nursery and Green Diamond for the seedlings that you guys donated to the study. So with that, thank you for your attention, and if there's any questions.
Is there anything in the literature about the growth rates of redwood and Douglas-fir seedlings in terms of the Doug-fir grew faster, right? Does that always happen with those species, or any other comparative studies like?
That's a great question. I haven't come upon. I'm not familiar, and I haven't done like an exhaustive literature search on this. This is still in its kind of natal stages. But I'm not, to my knowledge, I'm not sure. I would think that maybe redwood would typically grow faster than Douglas-fir early on, but I'm not sure. That's a great question.
Yeah, I was curious what type of seed stock it was, or what type of seedlings.
These were from the 092 seed zone. For the sacrificial seedlings were planted from Green Diamond seed zone 092, so our local seed zone.
What type of seed?
They were, I think the forest restoration class did, I think they were like, they were about, I'm not sure, they were about 2 years old, the seedlings.
Yeah, they were actually.
Yeah, okay. So they were 2 years old.
They're very, yeah. Thank you very much.
Okay, thank you.